Hello. We're continuing with my notes on life science and cell biology in a nutshell. We're learning about the different pathways which produce the different major groups of molecules called steroids. So, starting out with cholesterol molecules, cleaving of side chains via enzyme, hydrogenous and um, hydrogenase, dehydrogenase, hydroxylase. Uh, so various enzymes are used to produce the different the different steroid molecules. The mineralocorticoids is a group. Glucocorticoids, which are similar and overlap. Estrogens, androgens. So these are and progestogens. So androgens are largely in males, estrogens in females. Cellular locations of enzymes. So in pink, those enzymes are found in the mitochondria. In green, they're found in the smooth endoplasmic reticulum, which we learned about previously. So those are the different steroid hormones. So the mevalonate pathway, also called HMG-CO reductase pathway, begins with acetyl-CoA, ends with dimethylallyl pyrophosphate, which is used to produce the steroid molecules, as we will see. Let's see. We're trying to make DMAPP and IPP, isopenyl pyrophosphate. They donate isoprene units assembled and modified to form terpenes and isoprenoids, a large class of lipids which include the carotenoids, form the largest class of plant natural products. The isoprene units are joined to make squalene and folded into a set of rings to make lanosterol, which is then converted into other steroids such as cholesterol and ergosterol. So that's how you have the sequence of events in creating steroids. Oops. So mevalonate pathway starts off with acetyl-CoA. So these are the enzymes used: transferase, synthase, reductase, mevalonate kinase. So you're using ATP as energy source. So kinase, then pyrophosphate decarboxylase, isomerase, diphosphate synthase. So you're yielding. So you have the dimethylallyl PP is made, geranyl PP, farnesyl PP, from which you get squalene, which yields cholesterol, as well as ubiquinones, hemoglobinase, sterol, dolichols, geranyl PP, prenylated proteins. So sulfatases are okay, that was a different note. So that was everything that was my notes about steroids and how they're produced. it gets quite specific chemically. Um, so steroids are important. They're signaling molecules largely. They're involved in maintenance of sexual organs, as well as development and reproduction, as well as involved in the immune system, as well as regulating blood pressure and electrolyte balance. So they're pretty important. And the different kinds of steroids and vitamins are also kind of steroid. And my next note, so this is the main lanosterol is where steroids are they look like. So that's and it comes from the DMAPP, which was formed via the malvinate pathway, which we saw. So that's what the structure looks like. So mostly their steroids and lipids are produced in the smooth endoplasmic reticulum. Some of the enzymes are also coming from the mitochondria. The mevalonate pathway from acetyl-CoA. So dimethyl allopyrophosphate yielding cholesterol, etc. So another note, we were learning about lysosomes, which is another organelle. A lysosome is an organelle in the cell which is in charge of breaking down large macromolecules and parts of the cell, like even sections of organelles, or even um, bacteria or anything introduced into the cell that needs to be broken down. Lysosomes have a low pH of 5 because of a proton pump in their membranes, which reduces the pH. So we learned this previously. So there are various enzymes. An enzyme is a type of protein which is used for breaking down molecules and changing them. So there are various enzymes in the lysosomes with various functions. One of these enzymes is called the sulfatase. So here in this picture, lysosome is the red dots surrounding this cell, which is a dyed neuron cell. So lysosomes degrade organelles, membrane, or all kinds of different proteins, etc. So it's basically like a large vesicle or a bubble containing these enzymes which break macromolecules down. So inside these enzymes, inside the lysosome, you have lipases breaking down lipids, proteases breaking down proteins, nucleases breaking down nucleic acids such as DNA and RNA, and sulfatases. So this in green, you have another picture of lysosomes with their low pH, consisting of a lot of enzymes, acid hydrolase enzymes. They break down waste materials and cellular debris also. So here's showing the proton pump making the lysosomes acidic. So we'll continue and learn about this